Assalamu alaikum, this is Muhammad Hassan Shafiq, and today we will continue the ICB online series for grade 12 biology. We will be starting unit 17, which is nervous coordination, and the topics which I'll be talking about today are the coordination in living systems and the mechanism of nerve impulse. So, moving on, starting unit 17. Nervous coordination. The chapter has been taken from the following book of biology for Cambridge International AS and A levels, written by Mary Jones and Richard Fosbury, and all the images and text in this video have been taken from the following link. You can just click on the link and you can get the required book. So moving on, starting with coordination. Why is the living system coordinated? As we all know, are complex multicellular organisms containing of millions of cells. Different parts of the organism perform different functions, and it is essential that information can pass between these different parts so that their activities are coordinated. Sometimes the purpose of this information transfer is to coordinate the regulation of substances within the organism, such as the control of blood glucose concentration in mammals. नाउ अभी हमने लास्ट चैप्टर में होम्योस्टेसिस की बात की थी कि बॉडी के इंटरनल कंडीशंस को एक रेंज के अंदर एडजस्ट करना या रखना कितना ज़रूरी है. तो उसके लिए जो है ना, बॉडी के जो डिफरेंट पार्ट्स हैं दे आर कोऑर्डिनेटेड, राइट. अ सिंगल टिश्यू और सेल टाइप कैन नॉट परफॉर्म द होल फंक्शंस ऑफ द बॉडी. So that's why it is said that even within a cell, different organelles perform different functions. So different tissues, different organs, different cells perform different functions, but on the whole they are coordinated. Like we see quite a degree of coordination कि वो आपस में एक series में काम करती हैं. We see a flow of information from one part to the other, which is how the body works. So sometimes this change or response of the body is against a certain stimulus, and that is usually done to nullify the effect of that stimulus, or that stimulus can in other words also be a signal for like moving away from that particular part or region. सो बॉडी के अंदर एक रब्त है, एक बाकायदा हमें नज़र आता है एक सिलसिला, एक ऐसा जिसमें डिफरेंट बॉडी पार्ट्स आपस में लाइक कोऑर्डिनेटेड हैं, एक दूसरे के साथ मिलकर काम करते हैं. सो दे यूजुअली वर्क इन सच अ वे that the information they receive comes from the internal and the external environment. So there are receptors that detect stimuli. We will talk about this in detail in this chapter, that what are these cells. So the cells which have the uh, which can detect these changes are called the receptors, which can detect any change in the surrounding environment, the stimuli, and we will talk about these receptors in a little detail a little later. मैमल्स के अंदर जो कोऑर्डिनेशन है वो दो तरह से नज़र आती है. नर्वस कोऑर्डिनेशन जिसमें नर्व्स इन्वॉल्व हैं जो इन्फॉर्मेशन ट्रांसपोर्ट करते हैं फ्रॉम वन पार्ट टू द अदर थ्रू इलेक्ट्रिकल इम्पल्सेस, और जो दूसरी फॉर्म है कोऑर्डिनेशन की, दैट इज़ केमिकल कोऑर्डिनेशन, जिसमें केमिकल मैसेंजर्स कॉल्ड हार्मोन्स आर सेक्रेटेड व्हिच आर ट्रेवल्ड वाया द ब्लड. इस चैप्टर में हम नर्वस कोऑर्डिनेशन या नर्वस कम्युनिकेशन की डिटेल में बात करेंगे. कोऑर्डिनेशन जो है वो हमें प्लांट्स में नज़र आती है. इलेक्ट्रिकल इम्पल्सेस आल्सो मूव विद इन अ प्लांट. द रिस्पॉन्स इज़ रैपिड and different responses are seen within a plant to be effectively coordinated. We will also talk about coordination in plants at the end of this chapter, but right now what we will be focusing on today primarily is how nervous coordination is done and how the impulse or message is transmitted from one part of the body to the other through neurons. Nervous coordination, like the name suggests, this communication or coordination is brought about by the functional unit of the nervous system called neurons, and the mammalian nervous system is made up of the brain and spinal cord, which is collectively called the central nervous system. and the cranial and spinal nerves, which form the peripheral nervous system. Cranial nerves are attached to the brain and the spinal nerves to the spinal cord. Information is transferred in the form of impulses which travel along nerve cells at very high speed. Nerve cells are also called neurons and they carry information directly to their target cells. Neurons coordinate the activities of sensory receptors such as those in the eye, decision making centers in the brain, and effectors such as muscles and glands. So, you have talked about this. I'm sure you have talked about the major components of a coordinated action in your school classes as well. There are primarily five major steps, which starts from a stimulus. That stimulus activates a receptor, which transmits the information through a sensory neuron to a major control center, which in humans is a brain. That brain interprets the message and subjects it into subjective impressions through motor neurons, which end on effectors, which are either muscles and glands. A muscle then shows response by contraction and a gland by the secretion of a certain hormone. So the human nervous system, as you can see, it comprises the brain and spinal cord, which makes up the CNS, and the peripheral nervous system. As in this diagram, there are much more, but only the larger nerves are shown for your convenience. Neurons, the structural and functional unit of nervous system, each has a different function. There are three of them: the sensory, the intermediate, and the motor neurons. The sensory neurons transmit information from the receptors to the central nervous system. The intermediate neurons, also called the relay or connector neurons, as they connect the sensory and motor neurons, they transmit impulses from sensory neurons to motor neurons. So the information in the sensory is translated into another form of action via the motor neuron. Motor neurons transmit these impulses from the CNS to the effectors to bring about a response. Now, looking at diagrams of these, as you can see, there are three of them: the motor, sensory, and intermediate or the relay neurons. The motor neuron has a cell body at one particular type, then comes a long cylindrical cytoplasmic process called the axon, and at the axon we see the axon bundles. In the sensory neuron, the cell body is usually located at the center, and it is not as branched as a motor neuron is. While looking at an intermediate or relay neuron, it pretty much looks like a star, as it is oriented, its branches are oriented in all directions. At one end it will be receiving the stimulus or the impulse, and that impulse will be then transmitted through the other part of, or the other half of the relay neuron. So it's pretty much like undifferentiated as than a sensory or motor one, but as you can see, the degree of branching is usually quite high, and these are the major neurons which reside in the brain and spinal cord. Moving on, so what's going on? How are we performing our actions? The first is a stimulus, a change in the external environment enough to be detected. Like, it should have a minimum level for it to be detected by an organism, so that it can obviously show a response against that change. That change or stimulus is then detected by a receptor, which is usually a cell located in the sense organs. We have five of them, so we will have be having, it does not mean that we have five particular receptors. A particular organ can have even more than one specialized receptor type. Sensory neuron then passes the impulse to the central nervous system, in which present are many relay neurons. The impulse is then transmitted to the motor neuron, but before it is interpreted in the major coordinating center of the body. It can either be a brain or a spinal cord. Then that image is then processed and sent to the motor neuron, which carries the particular message for the effector to show its particular action. Right, the muscle is usually the effector, and the action of that particular effector is, in the case of muscle, a contraction, which can be seen by immediately withdrawing your hand after touching a hot object. Right. So on the basis of stimulus detected, we can have different types of receptors. We will be studying six of them over here: the mechanoreceptors, thermo, noci, photo, chemo, and osmoreceptors. The mechanoreceptors actually detect sensations of touch, pressure, vibration, hearing, equilibrium, the pressure of blood in the arteries, and the stretching of the internal organs. during functioning. Thermoreceptors detect changes in temperature, so these activate certain receptors which will then uh, adapt the body towards that particular hot or warm climate depending upon the temperature in the surroundings. Nociceptors respond to stimulus of pain, which can be by a chemical or physical damage to tissue. Photoreceptors detect changes in light intensity, and these receptors are usually present in the third layer or the retina of the eye. Chemoreceptors detect chemicals in mouth, for example, like they can detect taste, they can detect smell, and also body fluids. Osmoreceptors sense the osmotic pressure of body fluids. So, to put it all in a common pathway which is called a reflex arc, I will try showing you that how a coordinated action is done. The pathway followed by these neurons, or the impulse traveling through these neurons follow an impulse, which an arc which is called a reflex arc. So a reflex arc can be defined as the pathway along which impulses are transmitted from a receptor to an effector without involving conscious regions of the brain. So it actually tells you that how this information is actually transmitted from uh, a receptor, how it activates certain conscious regions in the brain, how those messages are interpreted and then sent to the effector for particular responses. The flow of information is the same which I showed you in the last slide. These reflex arcs usually have no intermediate neurons, like they do not touch particular conscious centers of the brain. They might have their relay neurons in the spinal cord in that case as well. So the reflex arcs, like, they might or they might not be involving the brain. So in that case where there is no intermediate neuron, the impulse might be passing the information directly from the sensory to the motor neuron. There are also reflex arcs in the brain, for example, those controlling focusing and how much light enters the eye. So for sensory perception of the eye, obviously the high light can damage the eyes in that case. So for sensory information to be interpreted, we might be seeing involvement of certain intermediate neurons of the brain as well, but apart from that, the normal responses or impulses or arcs which we see, usually like involuntary responses, do not involve very high conscious regions of the brain. So a reflex action is fast, it is automatic, it is an automatic response to a particular stimulus. The response to each specific stimulus is always the same. Reflex actions are a very useful way of responding to danger signals, such as the touch of a very hot object on your skin or the sight of an object flying towards you. So if I grammatically put, what I'm trying to say is that you can see that the receptor, a pain receptor, which in this case can be a nociceptor, is activated by the pricking of the finger, and that actually activates certain receptors which then transmit the information through the sensory neuron into the dorsal root of the spinal nerve. And in the case of spinal cord, we can see we have a central gray matter and an outer white matter. We'll come to that in a while, but right now we are just seeing this spinal cord as a major coordinator. This will then interpret and relate the information and send it through motor neurons into the motor and pellet or neuromuscular junction of a muscle, which will then be showing a power stroke or a contraction, which will result in the moving of that finger towards that stimulus. Otherwise it can be particularly quite dangerous. So this is how a reflex action is done. It is quite fast and automatic. So, transmission of nerve impulses, like we now do know that they involve ions, so it will be very rapid, right? There are these flow of information in the form of impulses involve changes in the disruption of electrical charge around the cell surface membrane called action potential, and we will talk about this in detail, that how an impulse is actually transferred from one part of the body to the other, whether it is sensory or motor. The information, how is it transferred from within a neuron, and also from one neuron to the other neuron as well. uh, for your information, let's start with resting potential. Our neurons are arranged or designed or born by nature in such a way that they have an electrical charge, or they have a potential difference associated with them, in a way that the inside of the membrane is negatively charged and the outer one is comparatively more positively charged. It's not strictly negatively charged, but compared to the outside, it's pretty much on the lower side. It's quite less positive, so we can relatively say that it's negatively charged, although it's not. So the outside has an abundance of sodium, and the inside has an abundance of potassium ions. This difference between the outer and inner membrane of a neuron has a potential difference between minus 60 to minus 70 millivolt, and this difference is called the resting membrane potential. Or we can also say that the electrical potential of the inside of an axon is 60 and is between something between 60 and 70 millivolt lower than that of the outside. So the resting membrane, how is it like maintained? How is this potential difference maintained? This is very important. Like, you should be knowing how it is done. So today I will be talking about the mechanism of nerve impulse, the conduction of nerve impulse in the body, and how this potential difference of 60 to 70 millivolt is maintained. So the first thing which is to be noted here, that on the membrane we have lots of sodium and potassium pumps. Now these pumps use ATP to pump ions against their concentration gradient, which means that sodium, despite uh, being low on the inside, is pumped from inside to outside, and potassium, despite being high on the outside, is pumped from inside to outside. Uh, so for every 3 sodium ions that are removed from the axon, 2 potassium ions are brought in. So for every 3 sodium which moves from inside the from the exoplasm or the cytoplasm of axon to the outside, so for every 3 sodium ions which the cell loses to the outside, 2 potassium ions come in. So the ions moving against the concentration gradient, like sodium is already low inside, so it's moving from inside to outside, and potassium, despite being low outside, is moving from uh, outside to inside. So both these ions are moving against the concentration gradient in a ratio of 3 to 2, and these pumps ensure that sodium is always more outside than inside, and potassium is always more inside than outside. So like you can see, we have a fine capillary glass tube with a potassium chloride solution, and when we connect it to the membrane, it shows a voltmeter reading of minus 60 to 70 millivolts, as you can see in diagram 15.9 uh, 2. Like, the second part of this diagram, you can see that the potential difference is now on the minus 60 to minus 70 millivolt, right? So there's definitely lots of things going on the membrane surface. The second factor which maintains this potential difference is the channels for potassium and sodium. There are more channels for potassium, by the way. These channels allow ions to pass through, not against, but in the direction of their concentration. Like, they move down the concentration gradient from high to low. So potassium, if it's already high, it will leave the cell and move towards the outside. So the loss of these positive ions, like potassium ions, will result in a more negative environment inside. uh, the third reason are the presence of organic acids, mainly, like, which are really having a negative charge if they are deprotonated. So they will lose their hydrogen ion in that case, and result in and provide a more negative internal environment on the inside. So these factors ensure that the inside of the cell of a neuron is always kept more negative than the outside. So the outside will always be carrying more positive charge than the inside. I'll diagrammatically show you that how actually you can like represent this. But what I want to clear you here right now is that sodium is always higher on the outside, and it has a higher charge density than potassium as well. So wherever sodium is more, the charge density of plus ions will be more, and obviously that environment will also be more positively charged. The inside environment like now comprises potassium. Potassium is not as high charge dense as sodium in that case, although it is electropositive. But since it is not as highly charged dense as sodium, so wherever potassium is accumulated or concentrated, the environment will be relatively more negative as compared to the positive external environment. So this electrochemical gradient, a double gradient like this maintained at a resting neuron when it is not conducting an impulse, is called an electrochemical gradient. So this is what I was telling you. Like you can see this tunnel shaped axon, the inside is negative and the outside is positively charged. Okay, and on the B side of the diagram you can see the pumping of these ATPase pumps hydrolyzing ATP and pumping ions against a concentration gradient. You can see sodium moving from the inside to the outside and potassium moving from the outside to the inside, right? Moving on, action potential. Now, how is an impulse generated, and if once it is generated, how does it affect the potential difference? So this is very important from an examination point of view, that what exactly changes are done and what's exactly going on. How is the membrane being disrupted all of a sudden, that this minus 60 to minus 70 millivolt which we were talking about changes all of a sudden to the plus 30 side. So this change is associated, the action potential wave or change is in a certain region of an axon is associated with the sodium ions being rushed in and the potassium ions being rushed out. So what exactly is going on here is the opening of certain uh, ion gated channels. Now these voltage or ion gated channels are sensitive to, if they are sensitive to sodium, the sodium voltage gated channels will open, and if they are sensitive to potassium, then only the potassium gated channels will open. But firstly and foremost, the sodium gated channels will open, which will allow the sodium ions to rush inwards. Once an impulse reaches the membrane or a particular region, sodium ions rush inward, okay, and the inside becomes more positive. Once all these gates have opened, and the opening of these gates follows a positive feedback. This is called a depolarization, since the deep like the polarity is now being disturbed in a way that positive ion was initially outside but now it's being accumulated inside, right? So this wave of depolarization is associated with the opening of the sodium gated channels, which are opened once the stimulus of a particular impulse reaches that particular point in a neuron. Sodium gates open, allowing all the sodium ions to rush inwards. Once all the sodium is concentrated inward, this is then followed by a wave of repolarization, which then allows the potassium gates to open, and that will be associated with the opening of the potassium ions from the inside to the outside, since now they will move down the concentration gradient and they were accumulated inside, so they will move from the inside to the outside. So initially when sodium is being rushed inwards, the polarity is being disturbed in such a way that now that minus 60 or minus 70 millivolt of the resting membrane is now changed to a plus sign, or the plus 30 millivolt, which is associated with the value of the action potential. So the polarity is dispersed or disturbed in such a way that now sodium rushes inward, which brings the inside environment more positive, and now the potential difference is on the higher side, which is plus 30. Right now this goes back to its original resting value once repolarization is done, and that repolarization is done with the pumping of the potassium ions on the outside, right, on the by the movement of the potassium ions moving outside, and that is done once the potassium gated channels open, and that is only done when the after all the sodium gated channels have closed now, right? So this is a stepwise event, it is not a simultaneous process. So if I can try to tell you what I was talking about in a graph, you can see that initially it was on the minus 70, then a wave of depolarization brings the action potential to a plus 30 value, which then comes down again to the minus 70 value, but it is then further gone down into the minus 80 region, which is like a hyper, also called hyper repolarization, as the potassium gates do not close as quickly as the sodium gates do. So this is usually associated with the losing of more potassium ions towards the outside, which brings a more negative charge called minus 80. So there is a time taken called the refractory period for the neuron to restore its original resting membrane potential value of minus 70. Now, like I just said, that there should be at least a minimum value for a neuron to conduct an impulse, right, or an action potential to be generated, and that for the threshold, the value in a human neuron is between minus 60 and minus 50. So this is the threshold potential. Anything below this will not generate a sufficient or a good action potential, or in other case you will not see any conduction of an impulse at all. So this is what I was trying. You can like read this, that first how the sodium ions open, right, and then the potassium ions open. The potassium ions are opened once the sodium ions are the sodium gated channels are then closed, and the opening of these potassium gated channels then allow potassium to leave the cell, which will result in a more negative internal environment. So the actual resting membrane potential of the cell is restored. Action potentials are discrete events, right? They do not merge into one another. Okay, the frequency of an impulse will be higher against a stronger stimulus, but the amplitude of the wave of depolarization and repolarization will always be the same. Like you will not see small and large peaks, you will see a uniform peak throughout, but obviously the frequency will be more or less depending upon how rapid that impulse is being conducted, right? So the frequency may vary, but the amplitude of the wave will not. The speed of conduction is much more when an axon is myelinated, which means that a layer of myelin is like a fatty lipoprotein layer is associated with the covering of the axon, which is secreted by a certain group of cells called Schwann cells. Okay, and these conduction in myelinated axons are much more rapid than an unmyelinated axon. Like you can see the difference of 100 meter per second in a myelinated against a 0.5 meter per second in an unmyelinated axon. The impulse is conducted from nodes to nodes, which are regions in a myelinated axon which are not covered by myelin. I will show the diagram in the next slide. So these are the regions where myelin is not there, and the impulse then jumps from node to node, which is called a saltatory conduction. And these nodes are at a distance of 1 to 3 millimeter on an axon, and the speed of transmission might be up to more than 50 times than in an unmyelinated axon of the same diameter. So you can see much more rapid conduction in a myelinated axon. Like you can see sprinting or jumping of an axon of an impulse on an axon from node to node, and these nodes are called nodes of Ranvir. These were the diagrams. You can see the impulse against a more stronger stimulus in A as compared to a weaker stimulus in B. So these are the nodes of Ranvir. You can see positive and negative charges. In an action potential, the charges are reversed, which creates a double circuit, and this double circuit then uh, carries the impulse from one node to the other, right? So the action potential therefore jumps from one node to the other, so we see much more rapid conduction. The direction of action potential is towards the left, and you can see opposite charges on two different nodes where the impulse is being conducted, and this is like behaving like a double circuit, right? Synapses are the regions or junctions between two neurons. The neurons usually do not touch, like the axon endings of one neuron do not directly touch the dendrite ending or startings of the next. Like there are 20 nanometer wide gaps between them, which are called synaptic cleft. So once an impulse reaches the synaptic knob of an axon, it then has to jump or like change its or alter its conduction process in a way that it has to travel through a gap now, right? So what exactly will happen? The let's say in this process of an synapse, we will call the presynaptic neuron of an axon and the postsynaptic membrane of an dendron, right? So the gap between these two will be actually, I will try to explain that how actually the impulse is being now chemically altered and being distributed from the axon of one neuron to the dendrite of the other. So if I diagrammatically show what I was trying to say, you can see lots of mitochondria, since this is a lot of quite an energy driving process. Once the impulse reaches the knob of an axon or the bulb of an axon towards its terminal part, it allows the fusing of certain vesicles towards the membrane of the axon, which then releases its content into the synaptic cleft, and those chemicals or signals are then bound to the postsynaptic membrane. If I try to explain this as a little bit more like in detail, let me say that let me try to explain a synaptic transmission in steps. Step number one is an action potential arises. You can see the negative positive charges, like the negative charges on the outside and the positive on the inside, since sodium is high now inside and potassium outside. The action potential stimulates the opening of voltage gated channels for calcium ions, which diffuse into the cytoplasm. Once these calcium vesicles fuse into the cytoplasm, they will cause the vesicles containing acetylcholine, which is a major neurotransmitter, towards the presynaptic membrane and fuse with it. The fusing of these vesicles with the presynaptic membrane will release the contents of these vesicles, which in this case is acetylcholine or uh, any neurotransmitter, into the synaptic cleft. This synaptic cleft will then bind to its receptors on the postsynaptic membrane, causing them to open sodium ion channels. Once sodium ions diffuse inward, the wave of depolarization is continued and the action potential is now ready to conduct an impulse in the next neuron. So this is how nerve impulse transmission is done from one neuron to the other. You can imagine the speed of this process, like it's 100 meter per second, so you can not even imagine how fast this actually is going on. So this was all about neural coordination. We will talk about the disorders, and we will also talk about the problems related to neural, like if it's not properly conducted, it can result to uh, major altered metabolic processes of the body. We will also talk about the effects of drugs on these neural coordinations. So this was all for this nerve impulse topic. I hope you got a good understanding. Please do read it from the book, and if you have any questions, you can ask me directly on the ICB YouTube channel. So this was all. That was all for this. I'll join you again in the next lecture. Take care of yourself, students, and it's Allah Hafiz from me.